Uma foto de novo. Hi, hello. Oh, yes. Yeah. So, the more you're satisfied, yes. I need to regret that you have the credit card. I would take anything. No, I mean, your photograph. I think it's a boarding pass. Maybe, could you share your I'm not sure what. Yes, I do. I'm not sure. Dia, senhor. Questionário que a gente fez para... Então, eu Mas você tem que...
Esse mesmo. Ah, deixa eu falar com ele. José. I'm unused to change. Vocês têm um acesso da página do ISM para fazer a configuração, alguma coisa?
Ah, não,
acesso, só o Walter. Porque seria interessante colocar esse link na página, o pessoal sabe qual a página e acessa para preencher. Porque como é muito longo, seria interessante só o cara só clicar ali.
Sim, pergunta pra ele se ele passar.
Você consegue.
Carlos, tienes que escribir para mí. Tienes que escribir para mí.
Ah, lo de ayer, sí.
Alguns desses computadores eles ficarão disponíveis tem um computador mas foi mandado para o e-mail de todos. Vocês têm um e-mail da
Sei.
Você tem? Você poderia então reforçar, então mandar um e-mail geral para todo mundo?
Mas então, qual e-mail? Porque esse e-mail, o último
Não,
e-mail do
o
Mauro.
link, por exemplo assim, esse link ele pode te mandar porque não tem nenhum programa para acessar, para colocar na página.
Não, então, mas
São zero.
eu não recebi esse link.
Não, você tá com a Mariana, daí pega com ela, só faz um copy cola e manda pra lista dos participantes. Que daí eles fazem pra. Mandamos
Ah,
primeiro
tá,
pra
não,
você.
mas eu não tenho a lista dos participantes, eu tenho a lista que o Walter mandou de...
Alguém tem a lista da Mariana?
Ah, ela deve ter.
Tá, então eles devem ter. Tá, então daí só pedir pra colocar na lista e mandar um e-mail pra eles preencherem naquele linkzinho ali do Google Docs. Beleza? Deixa eu falar com Obrigado, Márcio.
That's how you're going to see it, get to see everything. DNA opportunities in adults. We're learning this, so we thought that this would be a good opportunity for us to talk. We're going to have several presentations. The opening statements by Dr. David Brooks is going to give us some insights and some ideas to think about. And then Derek from University and team is here. There she is. Right in front of me. Then that guy with the coffee and water can Martin is going to present to us. And that will be after a coffee break. So I'll open our service group there. Brooks. Thank you. Once again, I'm not going to tell you what to do with monogenes, monogene DNA, because I'm actually interested in hearing about monogene DNA. But what I'm going to talk about is partly objective and partly very personal and subjective. And it has to do with the last 30 years or so of my personal activities involved with a lot of international biodiversity initiatives, almost all of which have failed. And so what I'm going to suggest is we actually have a really good idea of what the problem is and what we can do about it. But what we don't have is much support from the scientific community. And if you don't have any support from within your own community, then you cannot be an advocate outside. So let me just, and I think I finally figured out how to get the entire slide on the screen. At least I know it wasn't the first two. Well, it wasn't the Max law. It was one. Can you hear me, or do I need this? I can hear you. Many years of lecturing to undergraduates, so I'm going to project. Okay. So once again, there is this. One of the things that Eric Hoberg and I have been publishing separately and together for the last decade or so is trying to make the point that the biodiversity crisis and the climate change crisis and the emerging disease crisis are really different aspects of one thing. They're not three different, they're treated as three different kinds of business opportunities, three different kinds of revenue streams for academics, but in fact they're all part of one challenge for humanity. And that means that we actually, all of us actually have many responsibilities and very little time. There is a new major climate change assessment report due out at the end of this year. And one of the things that they're going to say is that the rate of temperature increase in the air has slowed slightly in the last 10 years. So it's not that the temperatures have dropped, it's that the rate of increase is slower. So it's a second order effect. And some people are saying that this is good news. It means that all of our recycling efforts are working and we're going to be able to reverse climate change. The reality is that because we're dealing with a complex system dynamics say that whenever a system slows down and pauses, it's about to do something new and drastic and unexpected. So in fact, we don't have very much time. We really need, if we're going to do something, instead of just waiting to see what happens. So, and I'm going to suggest that we are not trying to save particular parts of the planet. We're not trying to save, or we shouldn't be thinking, that we should be trying to save particular species. And I don't think we should say that we're trying to save our species. Because none of the scenarios, none of the models that have been done suggest that the biosphere is going to disappear completely from this planet. None of the models suggest that all the species on this planet are going to disappear, or that if we lose particular species, we're going to lose more than warm and fuzzy feelings about particular species that we have from our childhood. And none of the models suggest that Homo sapiens is going to disappear from this planet. There are too many of us, we're in too many parts of the world, our population is much too high, and there are too many of us who still remember how to catch food with bows and arrows. So in fact, Homo sapiens is not at risk of extinction. What is at risk of extinction is this thing, technological humanity. That's what we're fighting for, because every hardcore tree hugger and every hardcore anti-tree hugger absolutely depend on the internet and electricity and automobiles and trucks, field vehicles and GPS units and laptop computers and cell phones. And what we're trying to do, really, whether we admit it or not, what we're trying to do is to make sure that our great-grandchildren don't ask what kind of fish you catch with an internet. And we know from what happened in New Orleans after Hurricane Katrina hit, we know that the projections are actually correct, that at least in North America, when the power grid goes off, within 72 hours people begin killing each other for food in cities. That's how long most urban people can survive without technological humanity. More than 50% of the people on this planet now live in cities. So that's the scope of the problem. And that's actually the common ground for environmentalists and non-environmentalists. So we all have a vested interest in preserving this. Now, we as scientists have a responsibility to communicate how important this issue is to everybody, from our immediate family to society, including policymakers and politicians. But one of the things we need to stop doing is we need to stop making people afraid. Most of the Americans I've met who call themselves climate change deniers actually do not deny that the climate is changing. They're just afraid that if human technology has caused this crisis, human technology cannot solve the crisis. That's why they don't want to believe that there's a real crisis change event, because they're afraid. So we've already made them afraid. We don't need to make them more afraid. We need to take the words of Frank Herbert from Dune, and we need to say the litany against fear. Fear is the mind killer. Fear paralyzes us. Fear makes cowards of all of us. So this is what we need to tell people. The scientific community needs to be saying this. Instead of saying, watch out, there's a crisis, we need to be saying, don't be afraid, we have a plan. We cannot stop climate change, we cannot reverse it. Finally, Barack Obama is not saying this delusional stuff about stopping climate change and reversing it. Finally, he's talking about adaptation. His last speech, he finally said, we have to adapt. So far, only the Netherlands has actually done that. There's no other country on this planet that has a plan. Only the Netherlands. Evolution for us, evolution is the key element for what we can contribute. And that's because evolution is the only process that we know that can consistently restore a biosphere that has been damaged by climate change. This has happened before. That's why not only evolution, but especially paleontology is absolutely critical to our discussions about how can we anticipate, how can we adapt. Now, this is an acronym for a protocol that a number of us parasitologists have been trying to develop, document, assess, monitor, and adapt. At the moment, if you don't know the parasite fauna in a particular area, this is the stage that requires destructive sampling, at least for some period of time. But the rest of this, especially the monitoring part, with the application of molecular techniques like barcoding, is going to allow us to not only do this rapidly, but also do it with non-destructive sampling. And as some of you have already mentioned, conservation officers really don't like parasitologists. They want our information. They want to know about the parasites, but they don't want us to touch their animals to get the data. So this is a way to come to some sort of compromise with them. This is a protocol that's been developed over the last 20 years, beginning with the all-taxa biodiversity inventory concept. This is what the systematists of the world say we need to be able to provide the baseline. We need a clean taxonomy, including voucher specimens, names that are linked to voucher specimens in museum collections. We need to know how to find each species when we want it. We need to know as much ecological and behavioral information about each species as is possible, and we need that in a phylogenetic framework so that we can assess the historical context of stability and change. And we need all of that freely available on the internet. That was the impetus for the creation of GBIF, the Global Biodiversity Information Facility. What will this provide? Well, for us, it's going to provide baseline knowledge of our species in space and time, but putting it in a phylogenetic perspective also allows us what's called a retrodictive framework. That is, learning from the past to predict what happened in similar situations in the past. What can we learn from that about what's happening now, so that we can use the retrodiction from phylogeny to be proactive about policymaking, instead of just being very passive and saying, oh crap, it's still happening. And why do we need to cooperate? Why is cooperation part of this? It's because no single person or single research lab can do this for enough species, in enough hosts, in enough places, in enough time. There's also a critical role for training paraprofessionals, parataxonomists, and paraecologists in this, for a couple of reasons. One is we need more people, we need more hands to do this work. But in addition to that, the only way we can gain widespread public support for what we're doing is by linking our activities to socioeconomic development. Because 80% of the world's biodiversity occurs in countries where 80% of the world's poor people live. And those people are not going to preserve their local biodiversity if they need to sample it destructively to feed their children. Now, parasitology actually has a long tradition of recognizing this duality of public service and basic science. In the United States, the founder of the American Society of Parasitologists, Henry Baldwin Ward, was at one time the chairman of the zoology department and the dean of the medical school at the University of Nebraska. He left the University of Nebraska when the medical school faculty got together and decided that the dean had to have a medical degree. And at that point, medical parasitology and basic parasitology began to split apart. And that's one of the reasons that we don't talk together very well. I mean, there are not very many tropical disease people here right now, and none of us go to their meetings, and we're going to have to start doing that. Parasitology is special, and we need to talk about that. It's true that many of our species are pathogens of economic importance for humans, in agriculture, and in wildlife, and those distinctions are becoming less and less distinct all the time, those divisions between what's wildlife and what's domestic. Our species, our parasites, provide deep information about trophic structures in ecosystems and their changes in space and time. So, for example, What does this tell us as opposed to what this tells non-parasitologists? The non-parasitologist looks at this and goes, oh, God, you parasitologists do really gross stuff. And we go, let me tell you something about this picture. This picture says, this jaguar's stomach was full of big asterids. Not only that, but this jaguar was sick. See all this little granular stuff here? That means that that jaguar's stomach was in serious distress. And, you see this? The jaguar was trying to do something about it. And we can tell that just, this is the new version of the Greek religious leaders reading the entrails of animals to predict the future. We can actually tell a lot from the entrails of our animals. There's a lot of information we take for granted, but that's something that the public doesn't know very well. How much information we can tell just by looking at a worm. So, one of my favorite authors is Mark Twain. Mark Twain was famous, among other things, for saying that, asking the question in 1900, how do you know when a politician is lying? And the answer was, his lips are moving. Mark Twain also said, American democracy is the worst form of government ever created, except for all the rest. So he was a great believer that organized institutions were a necessary evil. And that it was actually up to individual people and groups of cooperating individuals to do the right thing, even if government wanted. This was his advice to the American Congress more than 100 years ago. And as some of you may know, the American Congress, the current American Congress, is not taking that advice. And most political bodies don't. But we have to do that. The problem is that, as most of you know, one of the basic principles of ecology is that when resources are limited, interest-specific competition becomes really intense. And those of you who have been applying for research grants or trying to get articles published in important journals know that we're in a period where there's intense intra-group competition. And that works against cooperation. We have to understand that that is what we're being manipulated into, and we have to try to resist that impulse. And against all of the outside pressures on basic science, we have to join forces, or we're simply not going to be able to get the job done, and we'll just be sitting there on the sidelines watching it happen. There is a saying attributed to Voltaire. I haven't been able to track it down, so Voltaire may not actually have written it. But there is a certain inevitability about inaction. And what I want to do, I come from a generation in the United States that says, I don't care how great the odds are, I'm going to keep fighting until I'm dead. And so that's sort of the personal part of this. I know why I'm doing this. And I think that there are a lot of individual benefits that can come from cooperation, but I also believe that without cooperation, we're not going to be able to do anything effective about helping to ensure some sort of stable future for technological humanity, which is going to include the future of our children, our unborn children. So, thank you very much. I hope that wasn't too depressing for the whole
I think we have plenty of time. If you want to make a question, Mike, I'll ask you two questions.
Yeah, I would also say that there are a number of articles about these, discussing these topics and this perspective that you can download from, under my name or Eric Hober's name, from the Digital Commons, from ResearchGate. I have a bunch of them on my computer. If you've got a thumb drive, I've got PDFs of many of these things. So there are these. If you want any of that information in a more detailed format, you can find it And then if you have follow-up questions, 6 months from now or something like that, I'm retired, so I have a lot of email time, a lot of free time. Just ethically. Yeah. Okay, great. Thank you.
So next, Dr. Merritt from University molecular approach for drug-active species recognition, advantage of biological species consumption.
Good morning, ladies and gentlemen. Thank you, Walter, for making me come here. I would like to thank all of you that you are willing to listen to us and people. Daniel, I am really wondering whether I have anything interesting to say. Okay, what I've done, with a pair of my eyes, I looked into the phylogeny of Pterodactylus from the point of molecules. And with the second pair of my eyes, I looked to them in a point of taxonomy. And the hypothesis is that molecular markers are useful for Pterodactylus taxonomy. Whether it's true or not, I hope you will decide yourself after the talk is finished. I utilize two markers. One is nuclear, the other one is mitochondrial. The reason to utilize only these two ones is that one is representative of the nuclear genome, which is inherited from both parents, so you can see whether the worms can reproduce or not. And the second one, mitochondrial, has got simply more resolution, so if you'd like to identify something really properly, you need this marker also to improve your thinking about the story, if you like to tell. Okay. What I've actually done, I treated these two markers differently. So I built the alignment of nuclear marker, and I used the pairwise deletion option. The pairwise deletion option lets you utilize the phylogenetic information as much as possible from the fragment of DNA that you are utilizing. From the second marker, I use different option. I use complete deletion option. That means that actually you utilize the phylogenetic information, which is included only in the left of the shortest fragment of DNA that you included into your analysis. Then what I've done, I actually created 3 trees. 2 of them I'm going to tell you some details. The third one is just a collective one, which connects these two trees mentioned. Why I did that, you already can see from the first slide that even if I divided the content of species included into analysis, none of them can meet a single name of a species included into analysis, neither I. But I'm not willing to let you, to make you reading all the names. What I'm going to show you in the tree which I call marine species. These marine Pterodactyl species are actually a species which are of a marine origin, but also purely freshwater, which came from the marine origin and are still, phylogenetic point of view, related to each other. What I wanted to show you in this tree is simply that there is this main division in the tree, which was already published in 1991 by British people, which divided the species of Pterodactylus in so-called species which have got short ideas and species which have got long ideas. So when you consider marine Pterodactylus species, you actually have taken into mind that they are in 4 groups, in 2 groups. I need that right hand. That these marine species are actually related to each other and have very little about this marine one, and I will show you later on that as well. Within this tree, I can see 8 lineages. As number, the number does not mean anything, just a number to communicate what we are talking about. And when we go deeper into the lineages, one of them is just a lineage which consists of species of Undulothrema. It's included here just to show you the dimension of the tree presented. The next lineage, the sixth one, is presenting species so-called Arcuatus-like. I would not like to tell you the details because actually the very well work done by Tine Huiste, and if you would like to ask questions about this lineage, I just direct you immediately to her and she will explain everything. But what I wanted to just show that, that the genetic distance, which is actually calculated from the whole region, so ITS1 is also included. Three are built on 5.8 s, sorry for the error, but the distances are calculated for the whole region, so that the shortest distance of the species included in this range is as low as 0.4, 0.7. And what is actually also interesting, let's look at within the species, the Gyrodactylus species related to each other comes almost from all continents, or all sea around all continents, which is really a great finding. When you go deeper into this, from the point of the taxonomy, when you utilize all isolates of a species, here namely Arcuatus, you can see that they form monophyletic clade, which we like, which we want them to form. And of course, then you can see from which countries they come from, so you immediately can have a point of what is the range of distribution of the species. The same is with the other species, so I just go fast with them. So when you see Gyrodactylus ostendicus, the same, it forms very nicely supported monophyletic clades with the species collected from these countries. When you go to Flavestensis, only two samples from Norway, but also from the point of taxonomy, they found very nice monophyletic clay. Gyrodactylus gonda, also from so many countries, very nicely supported. So fulfill the rules of the taxonomy. Gyrodactylus branchialis, the same, many countries, you can see the range of distributions of the species and forms the monophyletic clay. Okay, the only problem which is within this tree is Gyrodactylus cameroni, which comes from Canada, from Lapertus quadracus. The other host for the species, the main host is also Spiegelberg, it's fifth-time Spiegelberg, here it's fourth-time Spiegelberg. And I have to admit that I committed a crime, never committed a crime, because the sequence is shorter. And to utilize the sequence within this alignment, I just supplemented these with the sequences of the nearest relatives. And the nearest relative to choose was Gyrodactylus stephanus and Gyrodactylus arcuatus. And it's a very interesting case, because actually, when I supplemented the tree with the C at the position 32, which is the same as stephanus, or with the D at the position 32, which is the same as arcuatus, you can see how drastically the support is dropping down. So here the species support is acceptable, I would say about 70, but here 53 means that, well, there is something wrong with the species. But what is the conclusion of that? That we should devote a bit more time and think whether Cameroni is just simply not one of the strains of Arcuatus, which, as you can see, is distributed all over the northern hemisphere. I only included species which are deposited in GenBank, but also in our private collection. We know that it's in Iceland, it's in Far East of Russia, so it's everywhere on the free finance. Okay, next clade consists only from Gyrodactylus harengi. I am not going to go into the details of this clade, but the distance is already, which we earlier postulated, within interspecies variation of Gyrodactylus. And I think you know already knows the explanation. I had slides, but I removed them because there's not enough time to explain. But this clade needs to be worked out again, and probably something interesting comes out of it. When we go to the next clade, the clade which I can call Paranephrotus, I have to mention that already we have written in papers that dividing Gyrodactylus into subgenera is not visible by using molecular markers. I believe that. I wanted to present these to you, so I made a very nice alignment. I looked for the ends of the regions. I prepared everything as neatly as I could, and then finally you can see that support of Paranephrotus species is 100%. I'm not meaning Paranephrotus species by the protonephridial system. I didn't look at the protonephridial system, but the content of species here is what was called Paranephrotus. And I like also this clade because two of the species from Antarctica are included in them. The reason to collect this species was to just go as far as possible from Europe and collect something which is the most exotic to the European fauna, to enlarge this phylogeny. The conclusion is, well, they are very exotic. They clustered together with Norwegian and Polish Gyrodactylus species. So actually, this is the finding when we find that within marine Gyrodactylus fauna, the clades include species from all of the continents, and they have very much wider distribution than the fauna which is freshwater. And again here, just boring information, but you can see that once again the species are supported, so all the isolates of a particular species fits to the rule of the taxonomy to form a monophyletic group. Okay, when we go to the clade 4, from the first three demonstrated, you can see that they include species which are actually, they can be called Metanephrotus, and the same explanation. You can see that, okay, they are pretty well supported, and the same. I didn't check the protonephridial system of them, but the content of species is simply that's what was reported, that they are Metanephrotus. So there is something in it, and we cannot say that, well, we should just destroy the idea of subgenera within Gyrodactylus. And the distance of the shortest of the closest relatives is here, as it was reported in the paper, from 0.8 to 1.1, depends on the isolate you take for the calculation. And because at the same time when I was preparing alignment of this for this lecture, I did an inventory of all the species submitted already in GMAG. You can see that most of the DNA fragments used in the trees have got accession numbers, so they are just open for public. And of course you can find some submissions which don't fit at all, short sequences, old sequences. The same is also with yesterday what we checked with old morphology, prepared samples. When you blast this sequence, it will never show you Metanephrotus relatis. It will show you the Limnonephrotus relatis. This sequence comes from the host is Mainspine Spiegelberg, so I would expect that another European species which is from Mainspine Spiegelberg is Gyrodactylus puntiki. It is just a part of its DNA, deposited as Gyrodactylus rarus in Chile. Here, in this plate, again, the closest genetic distance of the species included is as low as 4.4, which is important information. And also, here there is an interesting part. You can see that I included some DNA sequences which are just in our collection. They are not yet deposited in gene bank. I just did it because I noted that this species with this accession number is very similar, if not identical, to Gyrodactylus pterygialis. It has got some mismatches, but very little, and I was wondering why the people were not naming this as Gyrodactylus pterygialis. The reason probably is because it's collected from a different host, so it breaks the rules of the host specificity, and because there were some mismatches, they didn't dare to name it pterygialis. I compared this to the sequences from Norway, and they were from Canada, and this sequence was also from Europe, so they didn't know whether the range of distribution is that high. I compared to other two sequences, Gyrodactylus pterygialis from Norway, Poland, from Kotitz, from I didn't write the name, but it was Gadus morva and also Marutus species. They are all identical, so I think the conclusion is that without any hesitation we can say that it's just the same species. And again, you can see quite a good support for the trees. Again, very fine, interesting finding that the Gyrodactylus alexanderi, which is a Pacific species from Northern America, was also by the Norwegian people found in Europe without any doubt. Okay, this is the case of pterygialis, and these are the Spolachis virens and Gadus morva, the host I was mentioning. Okay, the only problem which I already noticed within this is this clade lineage 8. The lineage is the sister lineage of freshwater Limnonephrotus group in Poland, and in this lineage there are two Gyrodactylus species, namely Gyrodactylus rugiensis and Gyrodactylus rugiensoides. You can see that there is lack of monophyly of certain species. It actually means that we have to work more on this species, and who knows, maybe there are just fish species instead of two included. I'm sure that already have got a solution for that, and we will talk it later. And also Gyrodactylus anguille is included in this clade. These people who work with Gyrodactylus, you know that it belongs to the subgenus Nephrotus. It's based on the special plant which was only seen in this single species of Gyrodactylus. The subgenus level was created for the species. It is perfectly included in this clade, so there is nothing special in this species, but no one was checking whether it has got this plant, so there is still an option that, well, it was just simply misidentified. The sequences come from all of the work that you can see, but not from the Baltic when the species was described. So we don't know, maybe on here there are simply more than a single species, and maybe the other one is just that one with this special plant. Okay, next tree which I created, these are the tree which I called African Gyrodactylus tree. These are all the species well done by Iva Chiprilova. I'm not going to tell about this tree at all. Again, if you have any questions, straight on to Iva, and she will explain everything what you are interested in. I just produced this tree in a reason that I wanted to demonstrate you that this lineage 8 from the previous picture is a sister group to a huge group of Gyrodactylus freshwater fauna from Europe, namely known as Gyrodactylus limnonephrotus. Here is the subtree. You can see this representatives of this lineage 8, then lineage which is called Limnonephrotus. Without this lineage, there is a Paganeri group, my favorite group, when Gyrodactylus salaris is included. And also here there is the out group of this Paganeri group, consisted of two species, Gyrodactylus gracili, hamatus, and Gyrodactylus rutilensis. You can see that support of the clades within different sets of species used are still very high, and the support of the sister clade is high enough to believe that they are really sister clade. Okay, when you supplement the tree with all the species known and all the, but not known, but all the species with DNA with is known and deposited in GenBank, you can see that already it's making a very well unreadable tree, and the sequences from the Paganelli group has to be combined together, because otherwise we could not read the tree at all. Here you cannot read the species content of the tree, but again, I can see that many lineages in the tree. 13 lineages, some of them are more interesting, some of them are less interesting. Let's go into and show what I think is interesting. Okay, then you can see that some of the lineages extracted from the previous tree, they combine the species from golden carp. So there are the species from a fish which is redistributed artificially by humans all over the world with the parasites on its body. So you can see that it was collected in many countries, and my comment to this tree is that all of the species are monophyletic, so they fulfill the rule of a taxonomical rule. The distances of them are like that, from 0.7, 0.0, and 1.3, so they are at the level of interspecies variation. It's a bit too high. You can see also from the shape of the tree, it's a bit too high within Gyrodactylus burlei, so I think that it needs further consideration whether they should become not more than single species, which can be decided when you include more samples and other markers also, which we don't have, unfortunately. Okay. There is another interesting clade which consists of species called Macronecus group. These are the species from European minnows with very distinctively different morphology of the hooks, and as you can see, they are deposited under two names, but definitely we can see that there are three species that's called Macronecus from Finland, which is actually from the type locality of the species, Macronecus from Czech Republic, and UC from Finland. This species, actually, all of the species were overlooked before the molecular markers were introduced to the field. Why they were overlooked? Well, my explanation is like that because they have got really different marginal hooks from the other 10 or 15 species of very similar morphology, we parasitologists, when we took them, we say, okay, this is done, I know it's Macronecus, let's come to this difficult one and try to distinguish them as well as possible. And that's why the group we know now that it consists from far more species in Europe than just in the past one, and now two, UC and Macronecus. Paperline, I don't tell much about the group. The group is well done by Eva Shkrilova, well selected. The only comment to that is that because it is created and created well on the sequences deposited in GenBank by the intraspecies variation and some other characters of the sequence actually shows that the sequences need further attention. The abyss files are deposited, they should be looked again and decided whether some reported variation is not just badly done. When there were very few sequences in the past, actually it was very difficult to run very high quality sequences. So there are people responsible for these sequences. I hope they do it again, and then this interspecies variation within this pattern group, but also within this other species will be corrected or confirmed that they are really true. Okay, and now I'm coming to the favorite part of the tree, vaganary, which is compressed still, and the outgroup. I'm just showing this outgroup because for the whole of my life, when the sequences appear, I was really convinced that these species are just synonym of the single ones. But again, I wanted to prepare this talk with a really good data set. I cleaned everything, I did it as best as I could, and you can see that the species names Geodactylus urtilensis, they are from roaches, but from roaches from freshwater, they became monophyletic. The quality of the sequence, because one of them is old, is different, but still when you look at the diagnostic nucleotides within the sequence, they are identical. And they come from Russia and Czech Republic, far distance. These two are identical, not these two, even the distance is closer. Well, I am confused. I would say, if I had to say and decide it right now, I would say two different species with as low distances of 0.2. So we are already within the distances which are normally known that they are intraspecies distances of Geodactylus within this group. Okay, when I expanded the vaganary, you can see how many species is already included. That was also a confusion because when I talked to Walter before the meeting here, we were a bit complaining that it's already almost 20 years of work with the DNA on Geodactylus, and so little was done, and we have to somehow make people work faster and clean the system. Well, we were wrong, Walter. A lot was done. I could not create the trees which can be placed on one table. There is a lot of information. All the talk which I am talking now to you for really long is based on this information. There is really a lot done. But of course, Geodactylus is a genus which consists of, let's say, 20,000 multiplied by 5 or 6 means 100,000 species. Well, let's leave something for next generation. Don't describe them all, because then we will be in problems. Okay, but still within the species you can see form assemblages. The trees are, as in Geodactylus, they are star-like. I didn't mention of this, but you notice that there are no supports here. We do our best, always try to make a phylogeny which is supported and which is resolved. Okay, if it is against the biology of the group, what are we doing? The biology of the group, I think, it creates such pictures that the species radiate. And why do you believe that they radiate in a manner that they come in from 1 into 2? It was already well done by Gina, showing that they come from a sticker break into many copies at the same time. Why knowing this way cannot we accept that then we will get trees which are not supported because there was not enough time to gather enough phylogenetic information to get the support of the branches. The same is with vaganary. Again, I would like to tell you some interesting part of the trees, not everything, because then I will give you with the information. Okay, so many assemblages I see in the tree. As I said, some are more interesting, some are less interesting for me. Let's go to what is the most interesting for me, of course. Salaris, the Mali problem. You can see that all the sequences follow the monophyletic tree. I am not going to say more about that because Jack Kolumna yesterday was explaining. But it's actually a good example, as I wrote here, of the violence to the taxonomy rules. We all in Europe, I have to admit, violated the rules, and I will show you how. For that reason, I need the mitochondrial sheet. You can see that Gyrodactylus salaris sensulatus, so this one which includes all the isolate of Gyrodactylus demari and Gyrodactylus salaris. And also an alien, you know about that from yesterday's talk, they form many lineages. I brushed the tree nicely so that you cannot see really brushy one, and then you can see how many. And in fact, Gyrodactylus salaris sensu stricto, we know that it's just this single lineage. This is the Baltic basin of Gyrodactylus, and this one, which is species specific to Salmosala. Gyrodactylus demari, on the other hand, is just this single trait. It is Gyrodactylus described from grading from Demalus, from Slovakia, from River Milet, and we know that it is this one. So what we've done in Europe, why I say that we were evaluating the taxonomic rules, some of us named these traits as Gyrodactylus salaris, this tree, and the rest as Gyrodactylus demari. The other of us do it like that. We claim that these are Gyrodactylus salaris traits, and only this one is Gyrodactylus demari. Well, I have to admit that from the very beginning we were wriggling as much as possible, never try not to make us write that any of these traits is Gyrodactylus demari. You can see in our papers that we always avoided making something because we never from the beginning believed that they, against the rule of host specificity, they should be named as two different species. For us, the only resolution is that all these mitochondrial traits from this point actually the gene pool of one single species named Gyrodactylus salaris, although it should be named actually, well, as you can see, demari, because there are far more traits of demari within this tree than salaris, but the rules of taxonomy are strict, and there is the rule of priority. Salaris was described sooner than demari, and well, then it is salaris. Okay, just to confirm this, that it is a single species, I ran as many as possible isolates of Gyrodactylus arcuatus. I brushed also the trees. You do not have to read. I was not supposed to prepare, so you read the localities, but even if the structure of these two species, global structure is the same for you, just looking at the shape of the tree, who can say that they are different? If I just remove this names here and split the tree, I'm sure that none of us would say that, okay, this is the other way around. They are just simply different, simply similar, and what yesterday was taught that the mean distance between this intertraits is about 3%. There are species with a bit higher distance, but the distance is too much talking. The distance between these is around 20, and it is just like that because of the nature of the market. It gets saturated. You will not get more information than you get. Remember that DNA consists only from 4 letters for nucleotides, and just a combination of these nucleotides will make that the mutations can come to the same place, and of course if they come, you cannot see any difference. Although the difference happened during the evolution. That's why there's the explanation that they look like they do. Okay, another case. What happened? Okay, I know what you mean. Later you will explain it. Another case that I wanted to explain. When we submit sequences to the gene bank, I think we should be responsible until the rest of our life to keep them updated. Because what happens is on the way we describe a species, we give it a new name, but the sequence rests in a gene bank with an old name like Gyrodactylus variability. When this sequence was submitted into the gene bank, Gyrodactylus the reality didn't exist, was not a strike, it was just misidentification done by a lot of parasitologists which found the species in European farms, but actually when the molecular markers came into use, we noticed that the species was described from Caspian region, and it's just completely, as you can see, completely different than this one. Okay, now everything is clean out. In paper it's visible. I think that everyone is convinced, but if you don't change the sequence in the gene bank, it still remains like that. If you ask a student to prepare a seminar, you know what he says immediately. He says that Urodactylus perianthini is exceptional species with very high intraspecies variation. Why? That's why. Because it really shows that. Okay. Another group quite interesting is the three species. Again, in some phylogenies, they are very closely related, as you can see, the intraspecies variation and intraspecific and interspecific is like that. So they are very difficult to resolve. But again, when you clean the sequences, when you edit them very well, then they all become very well monophyletic. The rule of the taxonomy is well fulfilled. And within this miniature, you also can see this what we call suite of the host. This is the Peptidae family, Salmonidae. This is Rogatensis, from which so it's copied there, and then you get again okay. A case of hybridization. There are more cases of hybridization in the tree. We find the most famous one in farms, namely Urodactylus lavareti. When I came into the business, one of the species which parasitize Oncorhynchus rainbow trout, Oncorhynchus mictus, was named and distinguished as Urodactylus lavareti. When we ran the sequences, then we immediately saw that actually the hybrid, because as you know, we are in favor of the theory that you are not allowed to change host if you are host-specific species without gene rearrangement. It's not like what we are saying without the first lecture done by Brooks, that they have time to collect the mutations, but they have enough time to recombine. And hybridization gives them sort of opportunities that's very fast. When you look into this tree, then you see that only within these two places there are mismatches, but two nucleotides within the sequence of, well, not even, but the long one, so means more than 1,200 are perfectly acceptable as an intraspecies variation. This tree is mitochondrial, so you can see how related mitochondria are within the hybrid here, and within the parental species, we call it this species, which is Pomeranian. There are two isolates here from Belgium and Poland, from Bart. The channel in Belgium is showing when I was there, I saw some roaches and happily they have this parasite, and the problem was solved unexpectedly, as many problems. When you can see that the distances of mitochondrial COX-1 sequences within Urodactylus as far as the intraspecies variation, so that's why the species, the mother of the hybrid is Urodactylus pomerania, and the father of the hybrid is Urodactylus lavareti. To make it a little bit fun, when you see the sizes of the webs, Pomerania is much smaller. So when you are a hermaphrodite and decide who is serving as the father and who is serving as the mother, it's done like this size matters. So, and the interesting point of this that, okay, within the hybrid farm, in Finnish farm, we didn't find identical sequence of the COX-1 from mitochondria, but you see that this sequence related of the isolate from Poland more than the isolate in Belgium. So actually, it's also logical. Poland is closer to Belgium, so we said it is mitochondria to be hybrid. Okay, not a single hybrid. We are currently working on the next one. These are really fresh. Just we got results really 2 weeks before the conference. We have got hybrid of Gyrodactylus peltis with a new species which I well not knew already because time is flying, but the species is right from France. You can see, thanks to Christopher, the sequences from Austria, Poland, and France form a really very monophyletic, identical sequences, monophyletic group. But we have also another sequence as a got by now only as an outlet of this hybrid, which is 1.5% different from Gyrodactylus thymali. Well, the predicted name for this species, there is such name, Gyrodactylus ordinicus, from previous Czechoslovakia. I don't remember, it was from, I think, Czech Republic, the locality. Well, I didn't name it. It's a task for Milan, when he sends his students to collect the species and find it as a homozygote, then I will just name it Gyrodactylus ordinicus. The morphology of the hooks are very similar. So the other option is, which I would not like to face, the other option is that Gyrodactylus peltis is just a younger synonym of Gyrodactylus ordinicus. What happens, we'll see in future. Okay, this case very shortly because it was explained yesterday, Gyrodactylus pannonicus cluster. I would like to look at it from my eyes with taxonomy and explain what it did. We actually, in our samples, and you can check the G-bank because they still are like that, had this sequence, and the species was misidentified, called the pannonicus, because very similar morphology, same host. Okay, why not we call it pannonicus? We do not distinguish it further with morphology. But then we got another one, similar looking from the same host, from different localities from Russia, and we wanted to decide which one is which. So the only way to decide it was just to go to Slovakia, collect the species from the type locality, and decide which one is of our sample is identical to this type locality, isolate. Well, the result, none of them. We end up with 4 of the species. This is real Gyrodactylus pannonicus from Slovakia, from the type locality. The others are just the way description. Okay. A warning for everyone, I've already got this lesson. When you tag species, try to tag them from the type locality first if you don't want yourself to run into such problems in the near future when someone does it from the proper way. Okay, here is a mitochondria, and you see that the species, the 4 species are also visible in the sea of mitochondria. They cluster together, but it's another story I not going to tell you. Like now you can see how the species look like the other, Solaris musculatus. And this what I told you, Lucy, so the distances you see twice longer than Solaris 7, but the species wide distribution from white sea, Baltic basin, and also on the southern part. So and the last case to explain, there are 2 names and 2 species now. Gyrodactylus clusterostei is a species from Philippine Stickerberg, an example of host-meat, and then you have Gyrodactylus apera, species of minnow. When the tree is done, again properly every single nucleotide is collected, properly edited, thought, then I end up with 3 groups. They group like that there is the White Sea localities, these are the these are the western localities, Finland, but also Czech Republic, and these are the eastern localities, Finland, because the division goes across, not across, but along eastern border of Finland and Russia. You can see that there are 3 very fine relatively well-supported groups. I already know, but the sequences are not such a there are more of them. This division between the east and the west in the Pterodactylus fauna in Europe is well known actually. Even the species which cross this border, they also always differ even with a single nucleotide. There is a division between this area. Well, 2 or 3 species, I would say 3. You may decide whether it is. Okay. I'm not going to read the conclusion. You may read it yourself. That's what I think. I think that even if DNA is consistent only from 4 nucleotides, so someone of you can think that has got 4 additional characters to the other characters which we are using into the species description. Well, yes, but each nucleotide from DNA has got its own position. And if you include this position, then you got billions of characters, because changing on different positions means something different for the DNA evolution. When you have a lot of characters, well, why not to use them when you would like to make a true story as true as possible? And the last paradox of Pterodactylus, thank you for your attention. I saw this picture of an old Pterodactylus, and well, again, from the point of taxonomy, am I Pterodactylus or Pterodactyli? Half of my life I spent as a female, and the other half as a hermaphrodite. This time. Okay, thank you.
We are a little bit ahead of time, a little bit. But we have plenty of time, so I have 45 minutes discussion, and we can have questions, and then we'll go to coffee break, and after coffee break, then we'll listen to Steve and Monty. And another guy going like this already back there. Giuseppe.
Thanks, Martin. Just a question about Bohemicus. Where did you get the material to sequence from? I mean, are you sure it is, because as far as I know...
This sequence from Bohemicus is not from the species of Everett, I will call it from the sequence which I got is one allele from the hybrid. We just prepared allele specific starters, primers, for 2 alleles which are in many copies, because it's ribosomal sequence, 2 alleles which are in the hybrid, and we could separate them. You didn't show, you didn't see, but you see that they are different in size, that much different, 1273, 1218. So when you cannot sequence them straightforward because they are index, so the sequence is destroyed. But when you amplify them as a separate allele from the same allele, then you can see enough and because they are one of size. That's the difference. When we find them in a world which is not a hybrid, and which I expect, well, the Bohemicus was described by Ergen from Paris, so okay, I would want to make a difference about that. I would say that it should be our European fauna species of the ground trout, which is a lost for instance. When we find it, then we can say that this allele comes from this species.
My curiosity is not, I mean, the sequence is not from the original material.
It's from the hybrid.
Okay, because, I mean, I had a look at the prototypes of Bohemicus, and in my opinion, my personal opinion, I think it's closer to Salaris than to this. Okay. My personal opinion.
Okay. For me, the morphology, well, we might decide, but in terms of a bit bigger books than salaries, the marginal salaries, which is actually characteristic to the heritage of the that was one I didn't do any analysis, I just looked at them and just maybe personally, okay, they look similar, heritage and stuff, but rather than say salaries, I see that. Still, we are looking for a pattern. In many cases, it's not easy to find. Yeah, we have a pattern. We have a pattern now because we have mitochondria, and we know that mitochondria. I didn't show you that we know that mitochondria are normally related to this 3%, but we are looking for the pattern of this type. I need a pattern. Okay.
What's your opinion on original hybrids? So original hybrids is the implementation of this reason for your explanation.
Human behavior is the reason. When we poison the worms, when we drag them in farms, they lost their chemo receptors to recognize each other, they try, and they succeed, they produce the hybrid. This is one reason why they appear in parts, and why the other reason is that we still believe, and we always notice that when the Urodactylus changes code, rearranges its own genome. And this is a way to rearrange the genome. So remember that Rainbow tried to take to Europe as eggs. It didn't bring any local North American Urodactylus source. And the other character of hybridization, even with the species like Theriathenoides or Zoote, is that when you look into the mitochondria of the species, you already have some. They are just one because hybridization and this genome rearrangement is very, very rare. Otherwise, how we could expect Urodactylus fauna is so high species specificity is rare. When it happens, because you can see it happen, it goes through really to the bottom, so it brings with itself the hybrid only part of the gene pool, which is the paternal and maternal layer. I think there is enough evidence for that, but there is a lot done.
More questions?
What is your estimate of the number of Chirodactylus species and how many ever day?
Well, there is something about 400 and named with the DNA and maybe 50 already because they come out. So this is what it is done with DNA. The estimation of the Urodactylus species was made based on the number of host species, the number of mostly fish, because they are mostly hosts of Urodactylus. That was 25,000. I am just making calculation for you. When Tina came to me and had just one aquatic species, we at the time, long ago in London, thought that it's just the same species of gobies. She did it nicely. Now you can see that there are six additional 25,000 multiplied by 6 with a better resolution is over 1,100 species. That's why I say we are closer to 1,100 species of Urodactylus than 25,000. 400 is something.
How many hybrids?
The mitochondrial tree and the nuclear tree are not consistent. When you look, you know something about that already. When you look at it, they are not consistent. Means that when these are the trees, the gene trees, the street trees are not that vicious trees like you see with the islands. Mitochondria, they are in the order of Solaris, but they come from another function. I think that through the exploration they went with the hybridization. And they swapped mitochondria, which are very old lineages. Probably all the roots of the lineages of mitochondria are older than but it is what it is.
That reminds me what Ambrose said yesterday, that the parasites are sometimes older than the host. The mitochondria are sometimes older than the host. And the mitochondria is just bacteria, right? And I think, so you agree that hybridization is much more common than we think in general? Yeah, not all of them are.
Well, Pterodactylus is specific, so I wouldn't really make it for the other groups. What I was observed, I observed that Czech people work with Dactylobulus, I think it's a different story in this one. So, but with Pterodactylus, I see that the mitochondrial trees, the nuclear trees are not lost, which is an evidence for the on the way to the evolution.
Any more questions? We can go to coffee break. We'll make it 30 minutes, and I'll call you back.
Quem sabe não deixa no lanche. Estava aqui. Elas vão sair, gente.
Deu um bom.
Thank you.
But it has to be for example, when you say I have a base, I have a sequence that's 1,000 bases, well, that's true. But each substance, each position has 4 objects. So each position in that gene sequence, by the time there have been 3 mutations, the probability of hole placement in the next mutation is 100%. So you have a lot more characters, but you also have a lot more confusion, enormous levels of multiplex. So there's this trade-off. More broad genes, because it represents coordinated, different genes coordinated together. There are fewer characters, but they're more stable. DNA is getting more information, but less stable. That's why total evidence is really the way to go. But DNA is expensive to collect. Easy to do. Morphology is easy to use. It's cheap to collect, but it takes a long time to become an expert. And again, you have to cooperate actively.
I participate in baseball. I talk about my first strike.
Well, I have a bunch of articles. If you want PDFs of it, are you going to drive? Yeah. Oh, yeah. Okay. Well, we'll just give it to me after the call.
Okay, that's okay.
Marina, só dia tô sozinha, Marina.
Tá vendo?
Não, é a verdade. Não parei.
Vocês conseguiram mandar e-mail?
A Letícia mandou.
Beleza.
Chegou pra todo mundo na lista.
Eu vou avisar para vocês da alfabetônica.
Tá fazendo ajuda aí? Corrige meu artigo. Não, mas o meu artigo só que... Mas é aquele formato, não é forma. É o verso? Ah, que bom! Não, não tem nada, não é igual, não.
Just give me a second. No.
Eu pensei voltar 15 para as 11. Porque antes era 11, agora
É 45.
Mas ela tá volta só 11?
É 11, aí 11h30. 11h30. 11h30.
Até meio-dia, mais ou menos. Se a gente voltar às 11 até 11h30, aí de 11h30,
Não, é 11h30.
daí...
As perguntas, sabe?
Tá bom, então a gente
15.
volta às 11.
I mean, we can't come back there. Because we had all the discussions. We gotta have the discussion. So many more questions. That means got to come to lunch.
11.
É pra minha mãe que a gente vai
Qualquer outra mãe, qualquer um. Entregar pro Paulo. Recibo de prazo fim de domingo, terça-feira.
Your little boy is beautiful.
Oh, thank you very much. His is that second.
minha mãe que eu vou no pai
I will try to make you.
My son is 15 weeks. He's already been to 2 conferences. I'll show you a picture.
Faz 2 dias que eu não converso com minha mãe. Eu e a Manuel. Eu espero ver se eu falo com ela.
Ok, agora eu vou editar o som.
Tá frio.
Meu avô não era. Oh, when you took the chocolate off
Assim, né?
Ela é tão mais bonita que eu. Não, eu pensei nisso. Eu vou falar, eu vou falar, não faz diferença. Deixa separado.
O Brasil. Começar a bolha.
Which
And then there's some questions that I couldn't answer. And I was like, okay, I think Walter will answer from the lab.
And I think it's really cool. Personal reflection. It all got raised. This I think is so we have this digital reflection. This is what the British metropolitan laws.
Nobody will check
Medo a gente ter, mas o Rio de Janeiro falou muito na aula do Carioca, a gente sabe receber Esse é um grupo de pessoas que trabalham no governo. Durante esse momento a gente fomenta a colaboração, nossa informação, buscas busca projetos mais imposição do que porque a gente está terminando o estoque do peixe tradicional e está indo mais muito de peixe. Esse é isso que a gente está fazendo. Quando é alimento, é uma das razões que a gente tem no Congresso todo mundo conhece o Rio de Janeiro, o que é isso. O Rio de Janeiro melhora a informação, vão virar uma boca muito rapidamente. A maioria das pessoas que queriam áudio são quase sinônimos. Os outros lugares eles vão aparecer também, lugares mas é quase uma exigência do grupo que eu sei que nessa cidade vai só olhar para lá de fora e a gente trabalha. carioca, curitibano, Rio de Janeiro é Rio de Janeiro. Gente carioca, maravilhosos lugares, culturais, típicos que tem aqui no centro de São Depende da cidade, vou lá, vou pra... Aqui é só a foto do Facebook. Só a foto do Facebook. Só a Hoje é sexta-feira. Você dormiu muito aqui em casa.
Não, eu não dormi.
É por isso. Mas é só no show de E ela tá com você, se ela fosse me agora. Não tem erro. Mas não tem erro. Cadê a... Você já viu me ensinando isso? Chata, é uma mala. É uma mala de madeira. Você está começando a me entender mais. I'm talking to a Scottish. I mean, actually, the Scottish is the same. Not trying to be nice, otherwise she'll put me in the corner. Let's go back to
Você tá meio guloso.
I know exactly how you're feeling. I went to Washington. Exactly. That's my bad. So I was using one of the t-shirts. And then it was told us to put the
Não tá gravado aqui? Tchau. Ela que me perguntou que horas é. Eu perguntei mesmo. Não. Ai que não, né, Maria? Tchau. Tchau.
Je te laisse finaliser ça parce que c'est toi qui es concerné. Je dis à la limite, comme je t'ai dit, si jamais il y a un blocage total et définitif, je ne sais pas pourquoi, sur ma personne.
Un valeur proposée de S4 un peu comme il est après il va partir dans la civilisation.
Genre 15 jours en France et après on déménage au Cameroun.
Et il ne répond plus à ses coups. Il devra
Non, mais pendant qu'on est là ensemble, on un peu de la manière un peu complète de faire les choses. J'ai lu le projet, mais je ne me rappelle plus. On voit exactement ce qu'on fait, est-ce qu'il y a des intérêts de faire des choses au Cameroun même, est-ce qu'il y a des intérêts de faire des choses au Cameroun, parce que c'est quand même des affluents qu'on combat. Donc ça pourrait être... Et par exemple, alors c'est pas un affluent du Congo, le Ntem est rempli de poissons qui ont fait disparaître les poissons locaux. Et si tu les disparu, tu ne trouves plus qu'une identité. Moi, quand j'ai cherché ces idées locaux pour regarder les paradis, plus que il faut monter dans les sous-petits machins. C'est aussi un bel exemple. C'est le bassin d'à côté. Le Ntem, il est sur le bord de l'Anse.
Donc, voilà. Qu'est-ce qu'on fait, comment on se débrouille, où est-ce qu'on fait l'info, est-ce qu'on fait la génétique, évidemment pas en Afrique, qui va faire quoi, est-ce qu'on prend des étudiants, lesquels, il y a tout un tas d'aspects, on voit règlement qui va faire 50% en espérant que ce soit la priorité, ça va être en bonne voie, mais on ne sait jamais. Tant que Gafal là, parfois j'ai les billets d'avion, moi à l'IRD j'ai l'argent, ils m'ont déjà payé, donc je sais que je pars. Et il te donne 2 mois de salaire d'avance pour t'installer. On peut t'acheter une voiture, 4x4 pour aller sur le terrain, comme ça on est obligé de commencer à y aller. Ma voiture, bon voilà, je sais que je pars. Allez, tranquillement, soit elle me dit au revoir, c'est comme tu veux. Tu vois, il y a déjà des règles.
There's a reason he gave the message. There he is. Anyway, some announcements. Unfortunately, we didn't get enough funding to support the special number of zoology on monitors. So we were more than welcome to apply to submit your bid and so on, but we couldn't get the funding enough to get a special number of journals. So we will be more than welcome to talk to individual people if you already have a paper. I think we can afford to support some publication in zoology, some papers and so on, but you have to talk to me first. Two important announcements is I think we are capable of supporting some of you, the cab, the transportation of some of you from the hotel to the airport on Friday and Saturday. But in order for us to organize this, we need you to go into our site, if you haven't done that yet, and fill the form, the online form, arrival and departure form. If you do that and you fill that until the end of the day, we will try to arrange you either in a bus or separate cabs with 4 or 5 people, 6 people. No. 4 people. There's a driver. You can get a fine because of that here. So we can organize you into different groups, either bigger groups for buses or small groups for cabs to take you from the hotel or from your hotel to somewhere else, to the airport, not to somewhere else. So where is our fun? We're not taking you to Brazil. And Andy and Marcus and Giuseppe O. Palladini, is the real Scottish of this group. We know that from the Facebook. Ask you to go online again. We have a computer in the back that you can use and fill the form of the questionnaire that we put online for Thursday roundtable. Okay, if you could, that would help us to try to organize the discussion. These are the announcements, and we are happy to have Dean with us. That's your first ISM, right? Good. Enough. And Dean is going to talk to us about molecular ecology of origins. Where are we, and where should we go?
Okay, so to start off, I've looked up molecular ecology, the definition. First on Wikipedia, that's my crime, but then I backed it up with BDS wrote definition. So what does it say? It's the field of evolutionary biology that is concerned with applying molecular population genetics, molecular phylogenetics, and more recently, genomics to traditional ecological questions. So this includes species diagnosis, conservation and assessment of biodiversity, species-area relationships, and many questions in behavioral ecology. So this is good to know when we start off this talk. So back in 1999, they started with the journal Molecular Ecology, and since then, actually the whole field changed, and it has been revolutionized by the advent of new technologies, and it's still changing today. So there's a paper out quite recently where they reviewed the current state of the field and outlined also some future directions. So this is interesting to have a look at. So what tools do they use in molecular ecology? Well, traditionally, they started off with mitochondrial DNA because it's very fast evolving compared to nuclear DNA because it's maternally inherited and there's no recombination. But nowadays, the more fancy methods include microsatellites and single nucleotide polymorphisms, and that's the nuclear genome that is being used. And microsatellites are short variable length repeats across the genome, and they're very species specific, but even specific to individuals. It's like forensics. You can really trace individuals with these markers. And single nucleotide polymorphisms, they are less abundant than the microsatellites, so you need many more markers if you want to have the same resolution as a microsatellite. But the good thing is, it's far more easy to define your haplotypes, because these are single base pair mutations, and the microsatellite repeats can be rejected. So that's why they now actually favor the single nucleotide polymorphisms. But then most recently, with this next generation sequencing, the field changed and answered the same old questions. So two examples. With this next generation sequencing, is when they look at two hybridizing species, now they can look at the complete genome, what's happening there. So here you see a comparison of two hybridizing species, and they looked at the pairwise differences across the whole genome. And what you can see is that the differentiation is not equal across the genome. So this allows us to have a test of the genetic view of speciation, as has been outlined by Wu. And the question there is, do you have speciation genes? Are there only a few genes involved, or is the whole genome involved? And when you have a look at this, there's already some direction that it's only certain genes that are involved in the beginning of speciation, at least. Another example, so with this next generation sequencing methods, you also need new bioinformatic analysis, because you have lots of data that now you have to make sense out of this data. And one of the problems, and this is especially in the case of parasites, is a problem of contamination, because you can have host tissues or endosymbionts, you have small DNA to work with, so it's difficult to get clean DNA. But now this is solved by separating, for example, in this case, metazoan and bacterial DNA in silico. So you sequence everything together, and then afterwards, there's new computational methods to really tease out all the different genomes. And what you see on the left is a nematode in blue, the genome, and then in orange, it's Wolbachia. So you have the two genomes. These are again whole genome reads of these species. And on the right, you see another nematode, and you see the composition of the microorganisms is much more diverse, and each color represents different species of microorganisms. So you have different disciplines, as I told you in the beginning. So phylogenetics, it deals with relationships between species. you can use phylogenetics to look at the origin of species. And if you go one level down, phylogeography will look within species and look at the relationship between different geographical locations. And this will allow you to reconstruct the colonization history of that species under study. And then if you go even one step down more, we're in the field of population genetics, and there we will really look within populations, and you can uncover or reconstruct the transmission dynamics within confined geographical localities. So we actually go from the macroevolutionary to the microevolutionary level. And in molecular ecology, we actually focus on everything what happens within a species. And actually, phylogeography is a bridge between phylogenetics and population genetics. So here we will look at the geographical distribution of genetic variation, because every species has a very confined geographic distribution, and this is influenced by the paleogeological and climatological processes like ice ages and drift, but also to some characteristics of the species, ecological characteristics, dispersibility. And so they define it in two broad categories: extrinsic factors and intrinsic factors. So this is how it typically looks like, a network analysis. Very wide sampling coverage is needed. And what you will see, so on this map you also see the coverage of the ice sheet during the last glacial maximum. And so this is a very strong force in defining the distribution of species. And we will come back to that later. So population genetics, there you look at genetic change in populations over time and space due to either mutation, drift, gene flow, mating system, and natural selection. So if you have a new mutation arising, what will happen with that mutation? It will either be fixated or eliminated. And this depends on the effect on the individual fitness and also on the population size of these species. And you have either advantaged, deleterious, and neutral mutations, and the greater the selection coefficient, the more rapidly the frequency will change, either to fixation or to elimination. In contrast, genetic drift, this will act on all mutations, and it will randomly fluctuate over time, especially in small populations. So the effective population size is very important here. So if we have gene flow, which is actually the term for successful migration, and with this I mean by a migration of the individual to another locality or population, and mixing so that the genes are passed and mixed in a new population. So during migration, many things can happen. So either you have a demographic bottleneck, if only a few individuals survive during migration, and in that case, your new founder population will have a very low genetic diversity. The other thing what can happen is that all migrants arrive safely, but natural selection will select for certain genotypes which are best fit to the new environment. So here the typical analysis are F statistics, analysis of molecular variance, to estimate your population differentiation, gene flow, and population fluctuations. And you can have assignment tests that really allows you to assign an individual to a specific population. So what do monogenes have to offer to molecular ecology? Well, it has been stated and shown by modeling approaches that parasites with a direct life cycle have a tendency for strong specialization and diversification. And the wide variety in the life history traits of monogenes and the reproductive modes and dispersal strategies give us an ideal model to test the effect all these characters on the evolutionary potential of species. They also usually have a short generation time, so this allows us to study or look at evolution in action. And it has been, I come back to pterodactyls, maybe more than once, my apologies for that, but they have been coined the drosophilids of the parasitic world, according to Bakker, Harris, and Cable. And I think they're quite right, because you can use them in nice experimental setups, and they have a very fast turnover. So there's many things that we can do with the monogenians in this field. And just to show that I also am interested in other parasites, we have polysomes, and there you can already see, even within this group, you have many different adaptations and characteristics. And all these are very interesting to look at what the effect will be on their genetic structure. And here I listed also, well, I copied the table of Walter's paper in 2003, and he listed all traits that could be important in adaptive radiation of the viviparous pterodactylids, but all these characters will also have an effect on transmission of the species, the effective population size, inbreeding, and so on. So there's many characters we can look at. So what can molecular ecology offer us, the people working on these parasites? Well, molecular markers can help us to look at parasite transmission, both the rate of transmission and the pathway of this transmission. It can also help us to study the mode of reproduction or the mating system. For example, in pterodactyls, is there sexual recombination or not? In how frequent is it? It allows us to reconstruct the epidemiological history of an infection or a disease, and it allows us to identify physical and environmental drivers of disease spread. And if we talk about parasites and infectious diseases, they have a new term, molecular epidemiology, actually to denote studies, molecular ecology studies in infectious diseases. And there you can see that you have different levels that you can look at the individual host, who infects whom, is there a difference between male and female hosts, between groups, between host populations or species, and then between geographic localities, which we call actually phylogeography. So what has been done? I listed a few studies, but this is by no means exhaustive. There are already quite a few phylogeographic studies, and I think one of the first are Pterodactylus salaris, which is no surprise given its devastating effect. And that was already back in 2003, I think, when they used the CO1 gene to look at population differentiation across Scandinavian populations. And there they found that the distribution of mitochondrial DNA haplotypes is consistent with post-glacial recolonization of the host. Then by Laetitia Plaisance, this is a study on two monogenians from butterfly fish in the South Pacific Ocean. There, I think 16 nests has been used. And what they found is, so there's two different species, and they're quite different in their genetic partitioning. So the first species, Haleotrema, you see it on the right. The number of pairwise differences is very low. It's only up to 9 differences, while the other species show a much stronger population differentiation across this scale. So they tried to explain this. So this unimodal versus multimodal distribution of the differences. And they came up with egg duration, which can vary between a few days to a few weeks, and the passive dispersion of these eggs with ocean currents across pretty large distances. This was the only way to explain the palmictic structure of Haleotrema, because larval fish are not infected by the parasite. I need to tell you that as well. Then another study where they used CO1 data could give some more information on the host specificity specificity of the parasite. And in this case, the CO1 revealed a lower host specificity than initially anticipated or expected. And one of the hypotheses was that this could be an adaptation to low host availability in deep sea groupers. So that it's actually an adaptation to survive in this kind of habitat. And then there's a study with a very long title, but there they used molecular tools to show that there's a lack of transmission between wild fish population and cultured fish populations. So they could show with CO1 again that there was a strong genetic heterogeneity between parasites from the wild and the farmed fish, and this actually contradicted the hypothesis of cross-contamination. Their next study, who recently came out in +1, there they showed actually the opposite. And here in this study, they looked at the planktonics from sea bream. And there you can see that they could, and according to the authors, the first molecular evidence of pathogen transfer between wild and farmed sea bass, sea bream, excuse me. And then finally, I also included a study, a phylographic study of some monogenians from the desert Chad, from India and Timbuktu, or here as well. And if you look at the CO1 network, there was no clear apparent structure. So they looked like one prolific population, a lot of migration. So this apparent high gene flow was here again attributed to passive dispersal of eggs and larvae, and also recent population expansion during the last glacial maximum. And based on the distribution of the genetic variation, which was lower in the north, they invoked post-glacial dispersion for this system. So what has been done on the population genetic side? Well, not that much. The first microset markers that have been published are from the group of Joe Cable in 2001. And there they, based on the microset library, they could isolate 6 or 7 polymorphic markers for Didactylus turimbuli and Ulaterudis. And all the microsets in Didactylus solaris appear to be monomorphic. So they were not useful in this study. So what did they do with these microsets? So in an infection experiment, they could show that there was sexual reproduction going on, 3 to 11%. In 3 to 11% of the infection, they saw actually genetic recombination, or they called it interspecific hybridization, between lab strains that have been in culture for many, many years. So very differentiated lab strains. And they could also show that these hybrids displayed hybrid vigor. So you see on this graph, in green, these are the parasite populations from the mixed infection. They reach higher infection levels, and in the graph below as well, compared to the blue line, which are infections which consist of only pure strains. And then yesterday you saw in the talk by Jaco Luma that they also used, well, or at least I think they had, if I'm correct, at least 13 microsatellites, and then they could see they were fixed heterozygous and usually 1 clone per host species. So their sexual recombination appeared to be very rare, and I actually wondered, he didn't talk about this, but did you see any evidence for the triploidy that you described in 2006 with the microsets?
Triploidy is very rare. There happens sometimes.
Okay, but you did see some triplets with microsets?
Yeah, few.
Very few. Okay. So that's something else what you can do with microsets to look at the triploidy level. So what can be done in this case? So some research questions are waiting to be tested. I just listed a few. What is the parasite gene? What's the unit of evolution? For example, what I think that in Gyrodactylus, the interpopulation, that this actually represents a traditional gene because you have recurrent generations of one host. But in case of oviparous genera, where you have the egg laying and the mixing of the eggs, probably the component population will be the unit of evolution. Then, is host switching more constrained in oviparous versus viviparous Gyrodactylids? What is the effective population size in the different groups? Do we see local adaptation, crowding effects on sexual reproduction, and so on? I guess this is the right place to add all the questions of the different parasite groups together. With respect to local adaptation, some other nice infection experiments have been done with Xenopus and Protopolystoma. And there, with these cross-infections, they could show some host genotype, putative host genotype, parasite genotype interaction, which is actually an indication for local adaptation and red queen dynamics. And in this case, genotyping of your parasite isolates would really help. You could do strain-specific experiments and look for this type of interaction. So for infection experiments, which I think is a very important part here, there's some very nice experimental systems like Gyrodactylus of guppies, on stickleback, Dolidoras, salmon, and so on. Also the tree frog and the polystomes. And I think we have very nice experimental systems. So why are there so few studies? Well, I think, oh, the figure doesn't come out. So I think, well, of course, which is very evident, is a lack of tools and markers. And this figure, which isn't very clear to you, but this is actually a figure of Tim Littlewood where he showed that, I will try to explain, that you have the flatworms here and the other parasitic groups, that you have a lot of variation within the CO1 gene region. So the forward primers, they don't align very well because there's many mismatches, both in the forward and in the reverse primer. So we had already some trouble with finding good mitochondrial markers that would actually work in all different groups. Now everybody has their own specific primers, which work, sometimes they don't. But it took a lot of time to develop these. And of course, if you want to do real population genetics, you need faster evolving primers, microsatellites or SNPs. And to develop these, you need a lot of DNA, and that's a problem with these tiny little creatures. And in the case of the microsets of Joe Cable, there was very low polymorphism. So what can we do about this? So with what we do with the schistosome neuracilia, they're like 200 micrometers, it's also very small. We collect them on FTA cards to fix the DNA, and then on these single punches we do whole genome amplification. And on this DNA, you can do complete exome sequencing, and you will have a complete genomic coverage by only sequencing the exons, of course. Or another method could be genotyping by sequencing. And this is a reduced representation library. So what does it mean? It doesn't require to sequence every base of the genome, but it will digest your genomic DNA with specific restriction enzymes. And then you will sequence all the fragments through next generation sequencing, so a lot of coverage. And then the polymorphism you find, these are your markers. So you don't need to design new primers, which will only work at for certain species. But this polymorphism is at the same time your marker. And now the run for one round, it's about €3,000, and you can sequence 96 samples, but you can have a pool design, and the prices are expected to go down every day, basically. So one of the last slides is the sampling design, which is very important. Just I guess, I think at least 20 parasites per population is like a minimum that you need. It would be good to have one parasite per host to avoid any effect of clones or whatever. The comparative approach is very important. So to pick out closely related species that differ in only one or two specific traits, which is doable with the monogenes. And then, of course, this controlled infection experiment that I told you about. So in conclusion, this mini review of phylogeography showed that there's a strong impact of the last ice age in the phylogeographic pattern of monogenes. The low differentiation, which has been found in quite a few studies, suggests that the dispersal abilities are much higher than anticipated, probably due to passive dispersal of RBRX. These molecular tools have been proven, molecular markers have been proven as a good tool to study host specificity and for demonstrating transmission between certain host species or between specific populations. And by combining nuclear mitochondrial DNA, hybridization events can be demonstrated, which is also an important factor telling you about reproductive mode and so on. For the population genetics, the application of microsets could indicate or demonstrate sexual reproduction in Gyrardactylus turbuli, only very rare sexual reproduction in Celaris. And the last slide for some future directions. There's already, in epigenetics, they already showed that there is some DNA methylation system in monogenian as well. I think they took a polystone for this study. So what will be the role of this epigenetic system or mechanisms in phenotypic diversity, in pathogenicity, and so on? I think there's a lot of to discover there. There's also been some functional genomic studies. They looked for microRNAs, or I think they're looking for microRNAs in Gyrardactylus celaris in Norway. And there have been some gene expression experiments and hooked gene studies on polystomes in Perpignan. So I think there's many exciting things coming on. And the monogenic community has everything, I think, to dominate the common issues of molecular ecology, which has a very nice impact factor, 6.3. So thank you for your attention.
We have time for questions. No questions? I thought you were going to make a question. Team Bob.
Yeah, very good. It's a big meal for the field. And I just think about the future of this research field. Because we see the different one base different, nucleotide different, and we normally treat it equally. But actually in different countries, it may have different meaning, and it have maybe in one position, one country means much more than some other positions. So
yeah, so they're not equal actually. One mutation is not equal to another mutation because it can have different effects.
Yeah, but the machine, we use the software, we use the machine, but the software doesn't know which country is more important. So I'm thinking about, I was confused when I did the research. So I've been thinking about, is it possible for us to really find the truth of this?
Well, one of the things which is also important always, especially if you're working also with protein coding genes like CO1, or you can always look for selection, whether even it's one mutation, whether it's synonymous, non-synonymous. But in the field with functional genomics, our knowledge is increasing about the specific genes, about microRNAs. So the more we know about genomes, the more we know what mutation might have which effect. So I think, I don't know what's happening to the genome of Gyrardactylus celaris, but that will help us a lot So I think it's important to really treat your mutations that you find very well and try to find out what's happening and not just treat it as some variation and to construct a network, but there might be some functional adaptation of that.
Any more questions? Just a comment. Amy, I saw your comment about Jarvaculus is the interpopulation of Jarvaculus is E15. We play around with ISSR in the lab, and it appears that it's very rare to find members of the same family within the same host. So it seems that they are really jumping around all the time. So they don't stay with their mother.
No, and in one of your experiments with corridors, you can indeed show that switching between hosts, individuals could also be a mechanism to avoid immune.
That's an old one. I'm talking about we use ISSR.
Okay.
And we try to identify members of the same family, the same lineage of virus.
Okay, you're looking at a higher level.
Yeah. We do genealogy using genetic marker. And we found one or two specimens of the same family on the same host, but they are spread all over the host. It's just, so I think they might be others.
because they have the possibility to switch as an adult.
That's right.
And it's true that indeed they might switch more than the...
Well, they probably switch the same. I don't know.
Well, that's one. I mean, that's what can be tested.
Yeah, it's great.
So your hypothesis would be it's the same.
I wish I had my percent.
But because I was a bit struck by the diversity in CO1, for example, in Giardactylus gondii in this case. It was pretty low. I expected more, but that's probably due to trade transfer.
I think you have a suggestion about the number of samples. You say that 20 for each, 20, for each one.
Population, 20 per population, one from 20 hosts.
Yeah, because I'm from with the ecology background. I always my students argue with me. They said, I read some literature. It is 20 is enough. But actually, for population, local population, if you only sample 20. I don't think it is enough for...
No, the more the better, but start sampling and then you will realize it's very difficult to reach 20 per population. So I put it as a conservative minimum, but I mean now if you really want to publish in molecular ecology, for example, you need sample sizes of 60, 80, 100.
Yeah.
But I think they need to be a bit less strict for parasitologists because it's such a tough job. So that's why I put 20. Demotivates. Well,
In your slide, you said avoid cornering. It's necessary to concentrate so that you also detect.
Yes, that's true. Yeah.
So it means depending per piece.
And I actually think it depends on the question you want to, the question you're looking at. Indeed, if you want to look at the population structure or you want to see whether there's inbreeding, you need to focus on a population from one fish and genotype all the parasites you find on this fish. But if you want to do some phylogeographic analysis, then I think it's better to have one person per host. So it really depends on the question you're looking at, and also at the marker you're using, whether you're using CO1 or the microsets. That's also.
It's a good idea to think that somebody can use samples later for any of those.
Yeah, that's another thing.
Tissue collection.
Tissue collection.
Thank you. We're still thinking about what you said, so that's right. Just doing. I would like to invite Joaquin then to trees. Some considerations of monogeny and phylogeny, and I put him in this job with this trouble.
Okay, thank you, Walter, for putting me in this trouble. I always come to conferences to learn from people older and wiser than me. And so, day before yesterday, we heard Dr. Brooks saying that never talk to an audience of specialists about their own field. And see me here talking about papers that you guys all wrote. Walter brought me in a very neat position here. So I want to say in advance to this talk that I will of course mention a lot of studies that people here in the audience perform, and perhaps suggesting some other ways forward or some mild criticisms. That's not to imply that whatever was written is not true, or just a little bit playing the devil's advocate, suggesting some new ways forward. Now, this is the outline of what I'm going to talk about today. So first I will give some examples of monogenic phylogenies to set the stage. Then I will try to consider whether there is or there isn't a clash between the outcomes of molecular and morphological data. Can we spot coevolution in such a way? Because it's also a big word here. And then saying, well, molecules, morphology, what kind of markers, what kind of data are we using nowadays for monogeny and phylogeny? And then a short final remark is, what do we actually use the phylogeny for? Now, a good way to avoid criticism of the specialist whose subject we're actually talking about is to start by people who can't disagree with you anymore, so that's why I will start with an overview of the 18th century monogeny and phylogeny. Now, since that would take us too long, I'll just mention that, of course, since people have discovered monogenians, they have set hypotheses about their interrelationships, their classification. For example, for a family many of us use Van den Eden, who suggested the family gyrodactylidae, which he was already concerned with how to classify those critters. Main reason actually why I mentioned Van den Eden is that he came from the same university as Tina and me, so until recently he was one of the better Belgian fish parasitologists. Now, if you see what kind of systematic basis do we use nowadays for to name monogenic groups, you see that many people actually still refer, or since there's not that much out there, to, for instance, to works that are already a couple of decades old, like the work of Bykowski, for instance, like Gusev's works. So, in a way, perhaps not that much has changed, or not that many people have been concerned recently with really naming new high-level new monogenic groups. I will mention about 3 relatively recent studies that try to make a comprehensive overview of monogeny and phylogeny. First one of that is, of course, the work of Malmberg in the 90s. And this paper, which you probably know, has, in my opinion, one of the big advantages of this paper is it makes monogeny and systematics quite digestible, quite palatable. It's a very neat overview of all the orders and families with all the characteristics. Malmberg uses the term of evolutionary capacity levels here. He has some quite explicit hypothesis, but probably the main criticism of this work is that there's no explicit system to infer the direction of evolution, to infer what is primitive or ancestral and what is derived, which in a way might infer or cause a certain risk of circular reasoning. I'm not saying that's the case, but there's definitely a risk of this kind of research. And as you know, much of experience, he also did it on a higher taxonomic resolution with the gyrodactylid family, mostly based on haplo characteristics and on the excretion system. But so the advantage of this is definitely that it very explicitly mentions a lot of characters that you could use, and that many people probably aren't using nowadays. A second example that I wanted to mention is Jean-Lou's work, which has probably the advantage as compared to Malmberg's work that it uses a very explicit cladistic method based on spermiogenic and spermatozoan characters. critique you could, of course, mention of that is that it only uses one character set, one data set as basis for this work. But on the other hand, for instance, I think this work teaches us that developmental characters like spermiogenesis can actually teach us a lot more than only looking at the structures spermatozoa in an adult worm. So this adds like dimension of developmental characters, so to speak. And then I guess the most recent work is the work of Walter and Kritsky, which is probably the most comprehensive in terms of character sets used recently. Sorry. And in terms of taxa included. Because I think also if you look at Malmberg's work, at Justine's work, you see that they use character sets that since then actually very few people have used. So perhaps that shows that there are a lot of lost arts in monogeneous systematics. A lot of character sets that no one actually looks at. And if you consider, for instance, the paper of Berger and Kritsky, I would also wonder, apart from the fact that there are, I guess, many lost character sets, how many people nowadays still work on such a big phylogenetic scope of monogeneous? How many people still work on that many monogeneans? So I guess both in terms of character sets and both in terms of taxonomic coverage, we are losing a lot of information. I, for instance, can say that I don't have experience with very many different genera of monogeneans. Now, since this is, I guess, more or less the most recent explicitly phylogenetic work on monogeneans that has tried to redefine our classification and our family level naming, so to speak. This is, of course, the one that has often been used for scrutiny of morphology versus molecules and for scrutiny of how we classify nowadays our monogeneans. And you see that, for instance, in the paper by Pete Olsen and Tim Littlewood, where they test molecular results, actually mirroring them with Walter's phylogeny, using, as the title of the paper says, a medley of molecules. Now, what are the conclusion of that paper if you reappraise the morphological phylogeny? Well, tree here is a reanalysis of Berger and Kritsky tree, but only for the families that were included in the Olsen paper. What you see is actually that there's insufficient overlap to agree or to disagree. There are a lot of missing data, a lot of families that haven't been covered in both analysis, and missing data where there are, of course, ways to treat missing data, for example, in a molecular-based phylogenetic tree. It's quite difficult to code missing data or to deal with missing data if you want to try the total evidence approach with molecules and morphology. Second problem is at which taxonomic level do you code your characters? Because the family level morphological tree was obviously so mostly coded all the characters on the family level. Whereas for a phylogenetic tree, obviously you use individual specimens, individual species, and you have sequence. And the third, so actually you could say there doesn't seem to be an enormous conflict between the moleculars and the morphological tree, as far as you can say. Now, there is a problem that we also see in recent phylogenetic reconstructions of certain family, like in the phylogeny of Capstalid by Lindsay Perkins. They compare it to the morphological traits in Capstalid, and you see that there are actually only 6 characters that you can plot on this tree, 6 characters that are variable enough but do occur throughout the family. And this is a problem that is actually even worse in Gyrodactylus, and you see that in the work of Marek and Jaco, that there are not enough discretely varying characters to actually make a decent morphology-based phylogenetic hypothesis within these families. Another problem, and that's a problem, for instance, mentioned also by Jean-Louis in a 98 paper of him about monophily of monogeneans, is that whenever we want to compare morphology and molecules, we see that we're actually focusing on different taxa. And I call this the odd ones out. For instance, if you see those two examples, Lagarocotyle, which is a salamander parasite, and Monsatskiella, which is one of the few intestinal monogenian parasites. Whereas they, I believe, have been raised to ordinal state, there's no molecular data on these, which is kind of strange, because you would expect every monogenian researcher to queue up the sequence those. On the other hand, Jean-Louis mentions in his paper on morphology, monophily, of monogenians, that whenever people try to reassess morphological characters, for instance, to look for synapomorphies, they often look at exceptions. And he mentioned the example of the eyes of a number of anabolic testicular but actually only looked at some very peculiar parasites like diplozooids. So I think one of the reasons why it's difficult to compare morphology and molecules is that for some reason or another, there's not very much standardization. We're not looking at the same taxa with both techniques. Something that is very prominent in all this work, and you see it, for instance, by me mentioning this special Lagarocotyle as a salamander parasite, is that, of course, we attach a lot of attention to the hosts, where do the parasites come from, and so we try to infer on this high phylogenetic level coevolution. So I'm not talking about no specificity level, but on the level of major clades. You see that, for instance, already in Malmberg's work, that he suggests that more primitive monogenians infect more primitive hosts, although he already has to infer some host switching at this level as well. You also see that in the later, in Justine's and in Berger and Titsky's phylogenies, that they infer some co-evolution and compare it to the iconic Fahrenholt's rule of more primitive parasites and more primitive hosts. Now, you also see that in case studies, like for instance the studies on Eusotrema, on Jagotrematidae, which are also amphibian parasites, salamander parasites, and also on this study by Benoit et al. So you see, if you focus on the monogenians that are on the non-fish hosts or the very primitive fish hosts, and that you would say, okay, there are very early divergences, synchronous with the early divergence of the vertebrate hosts. That might very well be true, and that's of course the conclusion that jumps out if you see papers like this. Now, there might also be, in my opinion, a bit of circular reasoning there. Again, I'm not implying that this is not true. I'm just trying to play the devil's advocate a little bit. How do you actually verify this kind of hypothesis? Is by assuming post hoc, if this were true, do you have a reasonable rate of molecular evolution? Because you actually calibrate it with the split of the host. And then I would say, okay, it might of course, probably it is true if you have a reasonable molecular evolutionary rate, but suppose we find on the big hippopotamus, like Louis suggested yesterday, we found a new monogenia, and it's situated here in the tree. Will we then assume that hippos are Precambrian, or will we say that we have to decrease the age of monogenians tenfold, or will we just say, no, it's an old lineage that recently switched to mammals? Obviously, we will say the latter. But why wouldn't that be the case also in these other levels of early vertebrate divergence? Just an idea. Now, why am I saying that? Oh yeah, this is a suggestion. If anyone ever gives you this kind of criticism on your dating of your phylogenetic tree, at the risk of being kicked out of the symposium, I found a good idea on Facebook to show that we can actually learn something from creationist people. Because what can you say if someone says that the Earth is millions or billions of years old? Just say, were you there? So if anyone of you ever critiques molecular calibration, just answer, were you there? Probably not. Now, why am I saying that we might have a problem of inferring coevolution on this level? Again, an example of Lisa Perkins' paper and PhD. On the family level of capsalids, you see that because they are so widespread throughout the fishes, it is clear that on this phylogenetic level you cannot detect coevolution anymore, because they have been host switches throughout the history of that family. So in that way, you would expect that if we add all the strange hosts, all the atypical hosts, so not only the reptiles and the amphibians, but also the cephalopods, the other invertebrates, we will probably not detect coevolution anymore. And in some way, we don't seem to expect that we can add the cephalopod monogenians in a tree and still expect coevolution, but we do expect to find that when we do that with fishes or amphibians or reptiles. So that might be a bit of a problem. Secondly, paper by Tina, we see that even if your rates of molecular evolution nicely correspond, if the evolutionary distances nicely suggest co-speciation, that Tina showed that actually in the scopies it might be differential hosts, which are much more recent events. because we don't only need relative data, we actually need absolute data to be absolutely sure that there has been coevolution. And the third problem, I think, with inferring coevolution on this level is, do we actually know what is the host of a certain monogenic nucleotide? And I give macrodirectives as an example, which has a quite pivotal role in Malberg's analysis, to say that it has the most primitive excretory system, so and it sits on a primitive host on a polypterus. But in fact, most macrogyros are on catfish, which are of course much less primitive than polypterus. So it might be a bit difficult if you find a monogenian to just say, well, this is typical of this fish clade or those clades. Now, some more technical remarks on what kind of data sets we could use nowadays. We see that some systems, as Tin already mentioned, some people succeeded in developing their own markers for certain specific monogenic groups, and so which increases the number of markers used, like histone 3, like elongation factor 1 alpha, etc., that you see in a lot of the works of Ian Whittington's group. So it kind of increases the number of monogenic markers available, you would say. But on the other hand, they seem to be quite specific, because these kind of markers are not really very much picked up by other researchers for other monogenic groups. Same goes for the recent increase in the number of mitogenomes that has been sequenced. Doesn't really seem to cause a huge increase in the number of mitochondrial markers that have been widely used. And even, as Tin already mentioned, even for the widely used molecular markers like CO1, we don't really see that they are easily being universally used in a wide range of monogenians. There keep being some problems. Recently you also see, of course, with the advance in mitogenomics, that other aspects of the mitogenome, gene arrangement, gene order, are being used. Is this the way forward? Well, I would think, of course, it will give us a lot of new information on deep monogeny and deep platinum phylogeny. On the other hand, as I mentioned a couple of slides before, we could say that it's actually more standardization, more use of the same techniques in a wider range of monogenians that we actually need. And I reckon that these techniques are not available to most monogenian research groups. Definitely not easy or cheap to replicate. So I don't think it would be necessary for all of us to start trying to sequence mitogenomes and look for gene arrangement. Now, drawing for some of my own work, and gyro work mainly, what could then be easy solutions? Many people have criticized the bandwagon effect you have in those couple of many used markers like ITS. Everyone has a gyro sequence and ITS of it and wants to make a phylogenetic tree, as I did as well. And of course, that is not the perfect marker for a phylogenetic tree, for deep phylogenetic influence. Now, on the other hand, I do believe that if you treat your multiple sequence alignment a little bit better, for instance, with some new software, you have to trim the alignment to look for very variable regions. Some software like that is Trimal and Tiger, to give us an idea. And that might actually seriously increase the resolution, like what I think the tree I just showed, a tree that is actually not yet contradicted if you use more, not enormously contradicted if you use more conservative markers. I'm not saying because I did it that this is the way forward, but I just think that in groups where you don't have a lot of markers at your disposition, that you can solve a lot by these treating your alignment to actually get more information, more conservative information out of it. Another thing, of course, that you can do is to combine, it's obvious, to combine markers with various resolutions. This is perhaps not a very good, this is an example from my own PhD where we combined ITS with CO1 and CO2 markers. So this is not a deep phylogeny, it's a phylogeny of some gyrodactyl species from gobies. But that does show that we can represent actually intraspecific, more or less geographic variation, and with it, between species variation in the same tree by using this combination of markers. Now, as I said, many people have criticized this use of a one-stop shop marker, like they have ITS, we try to do everything with it. This is an example I've drawn from colleagues of us who work in turbellarium flatworms dataset we reanalyzed in a recently submitted paper, and that mentions, of course, 18S as you know, is a marker for relatively deep phylogeny. But if you don't have many markers at your disposition, people working on topolarium is actually worse than for us. You see that they actually even perform relatively well if you want to distinguish species or cryptic species. So why not try to get as much information possible from the markers we have? And 18S is an interesting example because we see it a lot used in many monogeneans, but it hasn't been used that much in Trirodactylus. Whereas the paper with Eva, we could show that it actually performs very well to infer phylogenetic relations in this genus. And Kritsky also recently showed that in South American Trirodactylids, this marker does very well. These studies actually also show, both that our African and our South American studies, that looking at probably mostly overlooked haptolomorphological characters, additional sclerats and so on, actually shows that the discrepancy between morphology and molecules are not that much of a problem. I think it's mostly a problem of data rather than a problem of results of those data. Another thing that these studies might have shown that it is really a good idea to collaborate because we basically made similar trees on two different continents. And the last small remark I want to make is what do we actually use phylogeny for? Because we want to make these phylogenies and then compare them to old classifications to say whether a certain family is monophyletic or not. And something which struck me recently in a discussion with Andrea is that already a while ago people showed that heterocephalids are polyphyletic. Has been shown in study of Laetitia Plaisance, has been shown in study of Andrea, has also actually been assumed in Walter's phylogenetic work. And for some reason or another, many people, including myself, actually still mention astrocephalids as a family. Again, I'm not saying I don't agree, because I do myself as well. Why do we actually make phylogenies if we don't seem to change our taxonomic appraisal of these monogeneans? And the same might go for some paraphyletic genera like Trirodactylus. Do we want to split them, or don't we want to split them? I'm saying we have to, but for some reason or another, I have the impression that there's not very much feedback from phylogeny to taxonomy and vice versa.